Not sure. Okay. Okay. So, finally, ending the last lecture and sort of leading into what I want to talk about next is what makes a pathogen a pathogen. And what this comes down to is that we classify certain genes and code functions, like making a type 3 secretion system that will deliver effector proteins, toxins, all kinds of stuff that we've talked about so far. But these are virulence-associated genes. And also important is the organization of virulence-associated genes on the chromosome. So I sort of spoiled the surprise earlier talking about this, but what we find is that these genes are often clustered together. And so kind of refreshing us on what's going on is there this myriad of things that need to happen for a pathogen to cause disease, and that all of these boxes, we've seen various molecular functions that are associated with these things. So production of fimbriae, production of afibrillar adhesins to stick onto surfaces, the ability to form biofilm, the ability to deliver effector proteins into a host that modulate actin polymerization, for example, and induce phagocytosis, and potentially production of other toxins in addition to effector proteins. And then what strategies exist to compete in this host environment to ultimately lead to your success as a pathogen in your host, ultimately causing disease. And so things that we generally associate with pathogens are adhesins, so fimbrial adhesins, afibrillar adhesins, these examples that I gave you before. The dedicated secretion systems, so when we see type 3 secretion systems that are not a flagellum, we generally say that, like, yeah, that's probably associated with pathogenesis. Type 4 secretion systems, type 6 secretion systems. In gram-positives, if we find alternative sortases encoded in genomes, so they're sort of the housekeeping sortases that are just decorating functionally important proteins, sort of normal proteins on the pathogen, on a gram-positive bacteria's cell wall. And alternative sortases, remember that these are some examples that we saw for building fimbriae and papillae in gram-positive bacteria. And then far and away, one of the things that if we see them on bacterial chromosomes, we often are like, yep, this thing is a pathogen for something, are toxins. So if we find these AB toxins encoded on a bacterial genome, we can safely say that this thing likes to kill something, right? And extracellular enzymes, proteins, things like hyaluronidases, phospholipases, all kinds of stuff that would get secreted and degrade extracellular tissues, for example, are things that we would be looking for. And so when we find these genes, so I sequence a bacterial genome, I annotate it by comparison to other bacterial genes as best I can, and I find that there are some genes that look like pathogen, like toxins or something. And they're right next door to a type 3 secretion system, and they're right next door to another specialized secretion system that's probably meant to deliver this toxin outside of the cell, is that we'll often find that these virulence-associated genes are co-localized on genomes. So just like that maltose operon example maltose regulon example that I talked about before, the same thing holds true for these pathogenicity associated islands. Now, things that are interesting about these islands, and that this ties directly into this idea of horizontal transfer of genetic information is that they're often characterized by essentially not being there in non-pathogenic relative species. So a number of the really nasty pathogenic E. coli that are out there, if I were to compare them to the E. coli that's in my freezer here at UMSL and say, all right, what does each of these respectively have, is that the hemorrhagic E. coli is going to have some islands involved in causing intestinal inflammation and association with epithelia in various parts of my intestines, and are going to do some very different things than this kind of normal friendly E. coli. And interestingly, if we were to analyze the DNA content of these islands of genes, is that I would often find that they have a very different GC percentage than the rest of the genome. And so this is sort of a general descriptor that we have for bacterial genomes. So if we sequence them, I would just basically look at, all right, what's the ratio of G and C to A and T? And so for E. coli, it's around 50-50. For other bacteria, that ratio can be 60 to 70% GC, or in some cases, in a large number of cases, I would find that GC percentage is very low. Now, what's interesting is if I find GC percentages that are very different in one region of the chromosome compared to all of the rest of the genome, that's a really important signature. And that tells me that that particular region of the genome likely came from somewhere else. It did not co-evolve under the same selective pressures that the rest of this organism's genome did for hundreds of thousands or millions of years. Okay? These are generally, this looks like a recent acquisition into this bacterial genome. So, when we look at, say, the genome of the Salmonella enterica typhimurium that is in my lab, sitting in the freezer, what I would find, if I went and looked at its genome, is that we could see some interesting things. So what's marked here are in these sort of blocks around, that are marked around this sort of pictorial map of the genome, are these SPI islands, or like what we call Salmonella pathogenicity islands. And so if I were to go and look at what actually is in these things, so there's one that I've been interested in for many years, is SPI1, Salmonella pathogenicity island 1. And what's interesting about this is that it encodes that first type 3 secretion system that's involved in invasion. And nearly all of those initial effector proteins that we talked about that are involved in inducing actin polymerization, stabilizing actin, and then sort of relaxing this whole actin polymerization system back down to its ground state, right? And so basically everything that's required for that initial invasion event is encoded in this SPI1. All of these genes are packaged together and that they're co-regulated. And what's fascinating about this is that this island has a very different GC signature in terms of its percent GC in the DNA that's in that island compared to the genome, the Salmonella genome around it. So it's very clear that that came from somewhere else into the Salmonella genome. And when we look through these other Salmonella pathogenicity islands, there's Salmonella pathogenicity island 2, and this encodes the second type 3 secretion system that is involved in survival and basically remodeling that vacuole that Salmonella comes into. And basically, this is all the machinery required to turn that vacuole into a nice little house for E. coli. That's in SPI2. And so other things, when we go and look around at these other spies that are in the Salmonella genome, SPY4, for example, encodes a fimbrial adhesin essential for host cell adherence. Being able to stick onto epithelial cells to get in the position where the SPY1 type 3 secretion system is useful, those functions are encoded in SPY4. And just, if you go and kind of look at this, the publication associated with this particular figure, they kind of outline what all of these different things are. But these stars around the genome are actually basically inactive bacteriophages. And so, which really leads into where I'm going today, in that these bigger pathogenicity islands, some of them are actually just great big transposons. Some of them are mobilizable, kind of interesting plasmids that are able to pop out of the chromosome and reinsert themselves. And that these stars are all essentially viral infections into Salmonella. But these viruses had picked up over time and infecting lots of different hosts. They actually picked up effector proteins along the way. And so these are stably integrated bacteriophages that encode effector proteins that are substrates for the type 3 secretion systems encoded in SPY1 and SPY2, respectively. So how Salmonella became Salmonella is a really difficult knot to untangle, right? That there's a lot of pieces that have been acquired through evolutionary time for this pathogen, and that all of these different viral infections happened at different times. And if we're to go and compare all of the Salmonella everywhere in the world, I would generally find that SPY1, SPY2, and SPY4 are in them. But all of the rest of this stuff is pretty variable. And exactly what constellation of things lets Salmonella be a human pathogen specifically versus a porcine or pig pathogen only, or just being very zoonotic that's able to infect everything, kind of like what Typhimurium is able to do. These are big questions that are kind of at the forefront of pathogenesis in terms of like molecular pathogenesis now, and understanding basically what makes a pathogen pathogen. So it's one thing to be able to say, hey, we can identify a bunch of genes that seem to be involved, but how do all these things work together?
You said the stars are like inactive
Yep.
areas. So on the
Well,
one
they're
side,
inactive viruses, so they're phages, like phage lambda, will integrate, and it's basically stayed there so long that it doesn't actually pop back out.
well, because on the one it says, what do you call it, stop E?
Yeah.
As a, it has that, but then you have that on side one. So does that kind of say, like, invasion was primarily through that before the side one
Possibly.
was
Yeah.
put into the DNA? Or?
Yep. So it is definitely, now that we have all of this information, the chicken or the egg kind of questions that are out there, for sure. Right, so what order did these things show up? And how did this pathogen become so nasty? So one of the best kind of molecular sort of forensic examples that we have is actually Yersinia pestis, and that we've done a lot to be able to reconstruct what made that such a nasty human pathogen at particular times. And so, yeah, there are a lot of really interesting kind of genetic forensics that is out there in terms of reconstructing pathogen lineages. Right. So, and all of this feeds back into this idea of this pangenome and this core genome, right? That then if I were to say, what are some of the core things that I would find in Salmonella? Again, it's interesting that sometimes, for some pathogens, so Salmonella is it's based on looking at its core genome. While it might be a commensal in some avian species, in general, this particular species of bacterium, it is a hardwired pathogen. So this is what it wants to do based on its genetic composition, and then it encodes all of these things in just about every single Salmonella has SPY1 and SPY2. So it's essentially equipped to invade host cells and survive in vacuums. And so that is a core feature of what Salmonella does. And then when we look at all of these other species and look at these sort of pan-genome and sort of what things overlap and what things don't, is that these sort of rosette plots is one way that people like to represent overlapping genetic content between closely related serovars. And essentially it's just a big high-order Venn diagram. And what we can start to pull out of this is maybe, so for Salmonella Kentucky, are there, out of that 127 genes that are there, for Salmonella Kentucky, are some of those associated in, say, these yellow rosettes? Are these associated with the specialization of those particular serovars in infecting their particular host? And so this is kind of where we're using higher order comparative biology to be able to ascertain understanding what drives specialization of different pathogens. So are there any questions about this? That ends lecture 15. Yes. Okay. So we're going to get into as much as we can today about horizontal gene transfer, but we'll really spend a decent amount of time wrapping this up in the next lecture. So I definitely want to spend a decent amount of time on these ideas of genomics, because this is really where the field is going and trying to understand. We've ascertained a lot about molecular mechanisms, of different things that are in the arsenal of the bacterial world to be an effective pathogen. But understanding the origins of pathogenesis, I would argue, is equivalently important to understanding where pathogens headed in terms of their evolution and interaction with human hosts. So I'm going to give a brief review, and that might be where we get to today, about horizontal gene transfer to prokaryotes, and that it's a unique feature of prokaryotes, and that they engage in efficient and rapid and prolific transfer of genetic information across not only sort of subspecies, across species, across genus, across order. They transfer a lot of information across very, very disparate bacterial species, and this has profound influences on the evolution of pathogens. So there are three general mechanisms that transfer happens through: transformation, transduction, conjugation. And I will then kind of get into a little bit about mobile genetic elements, pathogenicity islands, and where we believe that these pathogenicity islands have come from. So how did all of these pathogenicity associated genes, virulence associated genes, how did they get packaged into essentially discrete units on bacterial genomes? Because their evolutionary history is far shorter than, say, the Maltose Regulon of E. coli, right? Maltose Regulon of E. coli has been hanging around in E. coli for millions and millions and millions of years. Whereas many of the pathogenic functions that some pathogenic E. coli have, humans have not been around, and many mammals have not been around for millions and millions and millions and millions of years. So essentially, how did these things rapidly aggregate together and become a functional genetic unit? And this is where these mobile genetic elements are really a major driver in the evolution of pathogens. So, because bacteria are haploid and they're asexual in the traditional kind of the traditional sense of generating offspring, they grow, they divide by binary fission, they're haploid, and so their daughter cells are essentially genetic clones of the parent cell. And so inheritance is strictly vertical in the absence of any intercession of new genetic information. So essentially, if left to grow and divide indefinitely, the only variation you would see occurring is through acquisition of mutation by spontaneous mutation, right? A random mutation. Now, what we do see that's very different from diploid sexually reproducing organisms is that we see horizontal transfer. Now, some of this is getting very muddy, especially when we think about the plant world, about this idea that we have vertical inheritance and that you have sexual reproduction, and that there's not really sort of gene flow between species, or at least it's very, very rare when this happens. The same genome sequencing that's given us deep insights about bacteria, for example, has also given us really deep insights about plants and animals, and that the sort of very strict concepts that we've had for forever thinking about the genetics of plants and animals, it's actually a whole lot murkier than I think we've thought for a long time. And so what happens in the prokaryotic world is that new genetic information arrives via horizontal transfer. So this is intercession of new genetic information in a sort of vertical lineage. And so what ends up happening is that for cells that have undergone horizontal transfer from of genetic information from another host, is that now they are fundamentally genetically different from their other siblings that were derived from this sort of vertical lineage, right? Now, where the processes of evolution come into play is that now this is a genetically distinct progeny from these, and did what the genetic information it acquired, is it beneficial? If it's beneficial in this particular environment, this particular clone will now increase in frequency in this population, and if it's a good enough set of things, say, what it acquired allows for resistance to penicillin. If I throw penicillin at this particular population, that clone will be the survivor, right? And so under stringent selection or hard selection, this is when acquisition of horizontally transferred information can be really, really beneficial for bacteria. And we can think about these in terms of like selective pressures we impose, like antibiotics. But for bacteria, the world in general is a terrible place. It is an awful, awful, awful place, filled with terrible, terrible, terrible things. I would not want to be bacteria. That's just, the world they experience, our world is pretty awesome, pretty nice by comparison. So they're under tremendous selective pressure all the time to be the best they can be all the time. And so through this sort of horizontal diaspora of genetic information, And rapid transmission between species is arguably one way you can generate a lot of variation that can be selected on by evolutionary processes. So, shown in that figure, there are 3 ways this traditionally happens, or most frequently happens. So transformation, transduction, conjugation. So transformation, I'm hoping, how many of you guys have seen this before in a microbiology class? Reasonable number? Okay. So I'm going to kind of hammer through this because I want to get to the good stuff. So transformation is essentially the ability of some bacteria to naturally take up DNA from the environment and incorporate that DNA into their genome. So not all bacteria can perform transformation, but if we look at many lineages of bacteria, E. coli included, so its evolutionary ancestors were to kind of reconstruct function, very clearly were able to do this. And so this is an old mechanism to be able to take up DNA from the environment and incorporate it into your genome. And so interestingly, this process of going and finding DNA and basically dragging it into your cytoplasm, this part, so we can kind of think of this as this sort of process here. So steps 1 and 2. This is something some bacteria will do, but not step 3. And E. coli, under really, really crazy conditions, will actually engage in 1 and 2. And so it has the machinery to take up DNA, it just doesn't like to recombine it into its genome. And one of the hypotheses that's out there, and I think it's a compelling one, is the origin of doing this is actually eating DNA as food. One of the things that happens is that to bring this DNA into the cytoplasm, and this is conserved across bacteria for natural transformation, is that if you're going to take up DNA, the sort of molecular pump to do it essentially hydrolyzes one of the DNA strands to generate the energy to run this pump. So when you think about DNA, it's a phosphodiester bond. Phosphodiester bond is a high energy bond. So hydrolyzing that bond is sort of like hydrolyzing a phosphate off of ATP to make ADP. Okay? So through sequential hydrolysis of one strand of DNA, you can bring in the other. So what you end up with is recombination of single-stranded DNA into the host genome, and then through DNA replication, that new sort of patched-in fragment of DNA becomes a fully incorporated piece of DNA in that host genome. And so this process in most bacteria is a very, very low frequency event. But there are other bacteria, very notably Vibrio cholera. So the causative agent of cholera is really, really good at this under certain conditions. And those conditions, it turns out, are when it's growing on a carbohydrate source known as chitin. So where do you guys heard of chitin before? Insects, right? And so where Vibrio cholera likes to live normally is in aquatic environments, particularly marine environments. And so it essentially lives on dead plankton and consumes them. And that's where it normally lives. And when it's growing on chitin, it becomes really, really naturally competent. So it really likes to take up DNA from the environment. And so this is one of the explanations that we have for the rapid radiation of Vibrio species in marine environments. And so if we were to go say, I care about Vibrio cholera, I'm going to study Vibrio cholera, the next thing you're going to hear from anyone that works on Vibrio cholera is like, well, which V cholera do you work on? Because there's a whole bunch of different isolates that people work on. They all have different properties. They have different things in their genome. Now, in and amongst the core things they do is that they all produce cholera toxin, But everything else is kind of willy-nilly. And one of the reasons that we have an explanation for this, the sort of rapid divergence of what these species do, or these subspecies of Viridia cholerae do, is that it's clear that they're probably rapidly exchanging genetic information in marine environments. Okay, so how this gets done is actually an adapted type 4 pilus. So the same type 4 pilus that was this retractile thing to attach to host cell surfaces and pull yourself in. Most natural competent systems use essentially a modified type 2 secretion system, where the outer membrane part is essentially using this sort of vestigial pilus of these apparati to essentially kind of retract out, and retract in, retract out, and retract in to drag this DNA through the outer membrane. And these interior components are some of the molecular motor components that we saw that ATPase connect to for the retractile pilus. Some of these same components are there, and they're essentially just hydrolyzing one strand of DNA to generate the energy to move that pilus piece up and down to drag the DNA inside the cell. Okay. So, are there any questions about transformation? All right. The other second mechanism that's really important is transduction. So transduction is a virus-mediated process in bacteria. And so bacteria, like us, like any living thing on Earth, there is a virus that infects and replicates in that way. Bacteria are by far no exception. The vast majority of viruses on Earth are bacterial viruses. They're bacteriophages. And so one of the things that happens during viral replication is it has to replicate its genome. And so for DNA viruses or double-stranded DNA viruses, they need to replicate their genome, and it's often in conjunction with and package that into bacteriophage particles, viral particles, and that packaging step is not perfect. Okay, so these viruses say, all right, I'm going to make 100. As a virus, I infect a bacterial host, and I'm sort of programmed to make about 100 of them. And 1 in 100 of me accidentally packages, instead of the viral DNA, packages host DNA. And when that happens, so 1% of this phage population, this viral population, is packaged with random pieces of host DNA. So these bacteriophage particles can float along and infect a new host. And most of the time, that new host is going to be a really closely related species or the same species of bacteria. But some phages have really broad specificity, and they're able to infect a lot of different hosts. And so you can deliver genetic information, that basically big chunks of host DNA that can recombine with a new naive host, and you can inherit function this way. The most important part of what happens with viruses in the evolution of pathogens is a process known as lysogenic conversion. So if you think back to that picture of Salmonella's chromosome and those kind of blocks and stars, all of those stars were bacteriophages that at one point in time infected Salmonella and got trapped. And so they essentially became lysogens, like phage lambda, and over time became inactive and basically just stayed there. So things that can happen for viruses, because they're constantly sort of infecting, lysogenizing, and then popping back out of the bacterial host and infecting new, sometimes closely related hosts, is that by hopping around, and we're going to see this when we talk about these mobile genetic elements, we kind of think of viruses as mobile genetic elements, is that mobile genetic elements in the prokaryotic world, they're really good junk collectors. So they're like the DNA hoarders of the prokaryotic world. They show up and essentially are just really recombinogenic pieces of tracks of DNA, and essentially they acquire toxins, they acquire effector proteins, they acquire all kinds of stuff from different places, pop back out, move on down the road, infect a new host, lysogenize, and hang out there. And essentially now this new host that didn't have these genes, things like toxins, now has a toxin. And so one of the really critical observations about the important role of viruses in the evolution of pathogens is that there are a number of really classic examples that lysogenization of a non-pathogenic relative by a bacteriophage carrying virulence-associated genes can convert a non-pathogenic bacterium into a pathogen in a single infection. And so this is incredibly important because Shigella, for example, I kind of mentioned that player a little earlier, that particular bacterium encodes a toxin, Shiga toxin. And so that toxin is encoded on a bacteriophage. So I can grow up Shigella that I have in my lab, I can treat it with some sort of membrane stressing antibiotics or treat it with ultraviolet light, and I can get that bacteriophage to essentially undergo its lytic cycle, replicate, package its genome that encodes that toxin. I can take that bacteriophage, infect my lab rat E. coli, and turn it into a pathogen instantaneously. So I can take that infected, just naive lab rat E. coli, I can now put it into a mouse and that mouse will die. Okay? Whereas I could feed probably grams of this normal lab rat E. coli to a mouse and it would be fine. So this is a really critical process, and it's very clear that this is what happens sort of routinely, especially later on when we start talking about emergent pathogens. So things that didn't used to be a pathogen, but all of a sudden are. Oftentimes it's because of lysogenic conversion. So this absolutely sort of instantaneous, in terms of evolutionary time, transformation into being a pathogen often occurs through lysogenic conversion. So are there any questions about this concept? Maybe you have two today. Okay. So pictorially, what I'm getting at is that there are bacteriophages like Lambda, which will infect and essentially integrate themselves. Under most circumstances, they integrate themselves into the host genome and become what's known as a prophage. And they're just a neutral passenger in the replication of this particular organism. And they just hang out. They don't do anything. Now, under very low frequency or under certain types of stress, you will induce these prophages into this lytic pathway where they package viruses and basically produce viral particles to go out and infect your hosts. Now, where does this lysogenic conversion become a player? Is that the genes encoded on this prophage are silent, so they're just not expressed until they're induced by a stress. So Shiga toxin producing E. coli, they basically carry a lambdoid or lambda-like phage called STX. This STX phage encodes Shiga toxin. The Shiga toxin is expressed when that phage undergoes, basically pops into this lytic pathway. And so one of the things that's interesting about Shiga toxin producing E. coli is that if you had a really nasty E. coli infection in your gut and you had basically hemorrhagic diarrhea, One of the things that can really, really be bad is if I wanted to say, I'm going to try and clear that gastrointestinal infection with antibiotic treatment. So Shiga toxin producing E. coli, if I did that, basically all of those Shiga toxin producing E. coli in your gut would start producing Shiga toxin. So what do you think would happen? It's bad news, right? I mean, you're going to be in bad shape and you may die. And so what often happens with these sorts of infections is that you're in the hospital. They would try and stabilize you, and essentially they're going to do non-antibiotic treatment type therapies to try and clear this infection naturally. Basically keep you healthy enough to fight off this pathogen, rather than treat you with antibiotics for basically the risk of inducing the production of really nasty toxins like Shiga toxin. It's what we're trying to do. So, with that fun, happy story, I will leave you today, and we'll hit it again on Tuesday, where we'll wrap up this discussion and have a few problems. Yeah.